We're going to talk about a very interesting topic. I think it's quite fascinating because it's very much at the intersection of two things. You're going to learn something that is very pioneering, and it's the intersection between clinical trials, which is the process by which our society creates new drugs, new treatments, and you're going to learn about AI, which is a topic that I assume most of you already know a little bit about. But the intersection between those two fields is creating, I wouldn't call it a revolution, but a huge step forward in how we develop drugs and how we ensure the safety of treatments and so on. So yeah, we're going to learn how we are digitalizing clinical trials with AI. Just a quick summary of what we're going to be doing is we're extracting clinical endpoints automatically from pictures. Clinical endpoints are the basis of patient safety, the basis of measuring the efficacy of a drug, and are the main thing that we want to measure in a clinical trial. So the ability to extract those clinical endpoints automatically with AI not only increases reliability, but also potentially reduces costs and increases the speed by which we develop drugs. Now, this is something that is very much in the intersection of two things, as I mentioned, clinical trials and AI. I myself am going to talk about the artificial intelligence part, more completely computer vision, which is the science of extracting data from pictures, seeing and then understanding with AI. But I have with me Diego Herrera, who is a senior leader in the company Almirall. He has been leading the efforts of Almirall, not only in drug development and compliance, but also in trying to digitalize, trying to implement new technologies into digital trials to make it quicker and better. If you could join me in an applause for Diego and can join us on the... Thank you.
Thank you very much. Thank you. Good afternoon, everyone. It's a pleasure to stay here. Yes, I work in Almirall. It's a Spanish pharma company that has a very long tradition in research and development of new medicines. Currently, in Almirall, we are mainly focused on bringing new medicines in the treatment of skin diseases, of skin disorders. And for that, we need to generate data. We need to generate evidence by means of clinical trials. In order to be innovative, we decide to implement digital connected devices in order to bring together the clinical research community and also the patients and be able to capture, transmit data at any time from any place. However, with digitalization, we can see that new challenges are coming, but also are opening more opportunities in the area of clinical research that I'm going to share in the following minutes. So, why we are talking today about clinical trials? I can find four main reasons. The first one is that digital clinical trials are essential in order to develop new digital medicine. So if you want to use a new digital health technology, then you have to generate the necessary evidence to get that technology in use for healthcare. A second reason is the study protocols requires a large volume of data. Study protocols are very complex, and protocols are requesting to collect multiple data coming from different electronic data sources. And this is huge effort, so we need to set up digital tools in order to be efficient in the way that we are going to manage all this complexity. But another important reason is because regulatory agencies are requesting every day more and more data that need to be captured directly from the patients. So for that reason, sponsors of clinical trials, we are investing a lot in order to set up systems where the patients can report their outcomes from any place at any time. And the fourth reason is we are now living really exciting time with accelerating solutions that are coming into the market, and then let me say that today the problem is not to find a solution, a connected device. The problem is to find in clinical research which is the solution that is going to fit the purpose of your clinical research. But what about the technology that, fact-checking technology that we are using in clinical trials? Well, I'd like to highlight the 5. The first one is mobile technologies. Mobile technologies are applications that can be installed in the smartphones, and these allow to the patients to use their own mobiles to report the data. Electronic consent systems is also a trending solution because it's allowing the clinical researchers and also the patients to manage. They participate in the consenting process electronically, including the electronic signature. Remote monitoring. This is very useful for monitors, study monitors, in order to review, verify, and also clean errors from the database in a remote manner. Wearables, sensors. This is also widely used in any phase of the clinical development. There's all these nanotechnology, microchips that allow to the patients to collect continuously digital biomarkers, and then today we have to wait for patients to come to the hospital to report their data. And finally, artificial intelligence is very useful in order to analyze big volumes of data, not only in clinical development, also in the process of drug discovery. So yes, we have big expectations based on all these technologies available. I would say that the first one is we can run what we say patient-centric clinical trials. And what does it mean, patient-centric clinical trials? It means that the patients nowadays, by means of these connected devices, can not only report their data, also they can receive interactive content, medical information that is going to be very useful for them in order to better manage the disease. And even more, they can receive instructions about the protocol, and they can be more compliant with the study procedures. The second big expectation, I would say, that is we are going to be able to capture more data, but not only more data, more robust data that allow us to generate new hypotheses, new evidences around the clinical development, so we can innovate the way that we are in clinical development. And as a consequence of that, we can reach another fair benefit or expectation that we are now growing, is the time and cost reduction. Because as we are capturing more quantity data in our clinical trials, then in the future, maybe it will be necessary to involve less patients and less sites because of this huge amount of data that is consistently collected in our study, so we can reduce cost and time. Now I want to focus this speech in the challenges that we have, in particular in derma disorders, in derma skills. Yeah, we can see that our clinical investigator has problems to quantify the number of lesions because the skin is a really large body organ. It can reach up to 2 square meters. So as you can imagine, the different symptoms and also the different lesions can be present in multiple locations of the body. So the physicians and also our investigator can have problems also to measure the accuracy of this treatment area where they have to apply the medication. We can see also that they have lack of standards at the time that they are doing the clinical observations. And the way that they can rate the intensity of the reactions to the treatment has a high variability. So the same lesion seen by 2 different persons can have a different rating of the intensity. And also, along the time, clinical trials takes longer because there is not just 1 week, but it can take years. So sometimes it's difficult to have a clear reference and precision when the treatment was allocated at the beginning and how it's evolving along the time. But for sure, with all these emerging technologies like artificial intelligence, developing digital photography is presenting a lot of new opportunities in the area of clinical development. So we can see two main group of opportunities. One is efficiencies. Efficiencies in the way that we are going to provide a tool, intelligent tool, that will allow the physicians to be more efficient in the way that they are doing the clinical observations. And also, we are going to increase the quality and the robustness of the data that we are going to capture during our clinical trials. So we can reduce the variability in the observations. We can be more precise in the location of the treatment area, because we are going to provide systems that allow to automatically count the number of the lesions across the body. So we are going to be able to save in time in this process. And the next one, what I mean, introduce novel endpoints. This is very important because we need to innovate. We need to make difference in our treatments compared to the previous one in order to give a new value to the patients. For sure, that we are going to have a more robust documented process. In clinical trials, if something is not documented, it doesn't exist. So it's very important to have very well documented process in a digital format where everything can be more traceable along the study. And last but not least, we are enabling teledermatology, that is something that is growing as a clinical practices, and also to allow remote visits and remote data capturing. And I think that this tool that Legit is developing is going to be a great tool that can support all these new opportunities in clinical development. So that is just time to give some explanation, details about how your technology is working.
That's good. Thank you so much. It is very interesting to me because this is something that we have, to some extent, co-developed. So the pharmaceutical company has these issues. It is very difficult to get reliable data, reliable endpoints. It's expensive, it's slow. So what we're doing with the AI is we are increasing, first of all, the amount of endpoints that you can get from one single clinical trial. And what does this mean? Imagine that you're in a clinical trial for, let's say, atopic dermatitis, and you only want to measure redness, right? So if you were to do that with investigators who are doctors, physicians, they will look at a patient and say, redness 3, redness 4. What's the problem? That if later on you want to measure dryness or desquamation or surface area, these are new endpoints, new clinical signs, new things that you didn't measure before. But because the AI looks at everything at the same time, extracts all the clinical signs that may be relevant, even those that may not be relevant for a pathology, here from a single picture. That's how we increase the amount. Now, I want to make it more concrete. So what does it do, really? So how does it work? I think about it as 3 features. One of them we call pixel to CSV. This means you have a picture of a skin lesion, and what the AI does is it gives you a CSV file, an Excel file, you can think of it, with hundreds of rows of data. And this is the data that you'll be using in your clinical trial. Now, the diagnosis that the AI gives can be useful, but mostly are feature of severity measurement. This is the most important part, because to measure if a treatment is working, oftentimes it's not only is it cured, yes, no, but rather how quickly is it improving in time. So you need to know how bad a patient with psoriasis is. Is it worse or is it better than 1 week ago? And the AI does that. Finally, which is just a funny feature that we have, quality control. So we have a lot of AI that looks at pictures and finds out if they are of enough clinical utility. We call that quality, but it's clinical utility. It is a good picture? No. In that case, you know right then and there, and you can prompt the investigator or the patient please take a picture again with better lighting, or maybe the area was not focused. So you can see how this increases the reliability and how this helps the challenges that we were previously mentioning. Now, we mentioned as well, there's this problem with the quality of the endpoints where different investigators in different days would say that the same patient is, they give it different scores. We call this inter-observer variability, what is between two doctors, and intra-observer variability, what is between the same doctor. So with the AI, we standardize this a lot. We eliminate intra-observer variability, that's done, and we reduce inter-observer variability by 13% in average. So this increases the quality of the endpoints. So maybe you can have a study with these patients, but statistically is as representative. Now, how is this done today? Most of you possibly don't know, but usually the way this is done until very recently is doctors would use scoring systems with pen and paper. I see someone who possibly is a doctor. So this is very tedious. So the score is for atopic dermatitis. This takes between 7 to 12 minutes to fill it. And it would mean looking at you and saying, okay, your redness is 12, the inflammation is 2. Now, this is very tedious. So what we have come up with as a society are the clinical digital calculators. The improvement is just that the calculation is done automatically. But again, it's the doctor who has to look at the patient and say, redness 2, and edema. What we're doing is we're doing this automatically with AI, where the AI looks at the clinical signs and extracts the intensity. And of course, then it applies the score. But in the end, you get the same endpoint, not only in 2 seconds instead of 12 minutes, but with increased reliability, and at the same time, the same pictures can be used. For example, if instead of this scoring system, you need that scoring system, maybe 2 years' time. This works with many conditions. So we're currently working with around 300 conditions, most of them dermatological. I mentioned psoriasis, I mentioned atopic dermatitis, but this also works with malignant diseases. So clinical trials with malignant or promalignant, like squamous cell carcinoma or even actinic keratosis, works with acne, even works with pressure ulcers, which are not this domain of dermatology. And you can see what the AI is doing here. It's looking at a picture and looking which are the clinical signs that are relevant to each condition. So with acne, it's looking at each lesion and counting them. With atopic dermatitis, it's kind of looking at the surface that is affected. With hidradenitis suppurativa, HS, is looking at lesions and then classifying those lesions depending on the type that they are, and then applying the scoring system. It's pretty powerful when you think about it. And that's how we get very detailed data. When you end up with study, you end up with a CSV where the data is very, very granular. You get week 1, picture 1, edema from 1 to 133. And that, let's say, in more than 100 rows of data. So it's very granular. So much so that you can maybe not repeat a clinical trial if you need to reuse the data. At, speeds. Obviously, I mentioned how a score takes within 7 to 12 minutes. If a person has to do it, we are using a very valuable resource because people are very limited, and especially dermatologists. Dermatologists are very, very, very limited, and the little time they have, they need to be healthy patients. So we ask them time of their lives so they can help us develop drugs. But if we can do this quicker by helping them with something that is automating part of the tedious work, well, what's not to love? Quality control. This is a data. 10% of the pictures that doctors take are invalid directly. So if you would just use the quality check, you would increase by 10% the validity. And I think that's pretty much everything that we had to say. We had like 9 minutes, but I think it's important in case you have any questions. either about the AI or the clinical trials, we're happy to answer and turn this into a discussion. You don't have to be an expert. No question is stupid. All question is possibly good to start a dialogue. Anyways, that's all from our side. So thank you very much for being here, and thank you very much for listening.